T helper cells differentiation. T helper cells, or Th cells, are a type of T cell that play an important role in the immune system, particularly in the adaptive immune system. Mature Th cells express the surface protein CD4 and are referred to as CD4 positive T cells. These cells suppress or regulate immune responses through their capacity to help B cells make antibodies, to induce macrophages to develop enhanced microbicidal activity, to recruit neutrophils, eosinophiles, and basophiles to sites of infection and inflammation, and, through their production of cytokines and chemokines, they orchestrate the full panoply of immune responses. Following T-cell development, matured, naive CD4 positive T cells leave the thymus and begin to spread throughout the body. Like all T cells, they express the T cell receptor with an affinity for class II MHC, and CD4 is involved in determining MHC affinity during maturation in the thymus. Class II MHC proteins are generally only found on the surface of specialized antigen-presenting cells. Helper T cells become activated when they are presented with peptide antigens by MHC class II molecules. Once activated, they divide rapidly and secrete small proteins called cytokines that regulate or assist in the active immune response. In order to be effective, helper T cells must determine which cytokines will allow the immune system to be most useful or beneficial for the host. Activated CD4 positive T cells can differentiate into one of several subtypes, including Th1, Th2, Th17, Th9, follicular helper T cell, or Treg. Signaling from the APC directs T cells into particular subtypes. These subtypes secrete different cytokines to facilitate different types of immune responses. Th1 cells are the quintessential cell type involved in cell-mediated inflammation and delayed type hypersensitivity reactions. They are most often defined by their production of interleukin-2 and interferon gamma, but have been reported to produce a number of cytokines, including tumor necrosis factor, lymphotoxin, and granulocyte macrophage colony-stimulating factor. The signature cytokine of the Th1 subset, interferon gamma, is a potent pro-inflammatory cytokine, which has a number of important roles, including increasing the expression of Toll-like receptors by innate immune cells, promoting IgG class switching, increasing MHC1 and 2 antigen presentation, induction of chemokine secretion, macrophage activation and increased phagocytosis, and is involved in several autoimmune diseases. Th2 cells are recognized for their role in host defense against multicellular parasites and their involvement in allergies and atopic illnesses. Th2 cells are best known for the production of interleukin 4, 5, 6, 10, 13 and 31. Interleukin 4 is a multifunctional, pleiotropic cytokine. It promotes the survival of lymphocytes, differentiation of dendritic cells and anti-inflammatory response in autoimmune diseases. Interleukin 5 and 13 are two other major cytokines produced by the Th2 subset. Interleukin 5 was initially described as T-cell replacing factor, which is secreted by Th2 cells to stimulate antibody production from activated B cells, and it also enhances proliferation and differentiation of eosinophil precursors into mature eosinophiles. Interleukin 13 is known to inhibit the production of pro-inflammatory cytokines, including interleukin 1 beta, interleukin 12, and tumor necrosis factor by monocytes, promote protection from Th1-mediated pathology, and it also serves as a B-cell co-stimulator that facilitates B-cell activation and maturation. Th17 cells are involved in host defense against extracellular bacteria and fungi infections. Much of the pathogenic functions of Th17 cells have been attributed to the secretion of interleukin-17, which promotes pathology and enhances inflammation and tissue damage. Interleukin-9 has been identified in a subset of T cells distinct from Th2 cells, now delineated as Th9 cells. Interleukin-9 is known to promote survival and enhance suppressive function of Tregs. They are also important in promoting mucus production and activation of mast cells as well as eosinophiles. Follicular helper T cells were not initially described based on cytokine production and transcription factor expression patterns, but demarcated as a unique population of activated CD4 positive T cells in the germinal centers of human tonsils that express the chimokin receptor 5. TFH cells provide help to B cells for the generation of germinal centers and long-term protective humoral responses. A subset of Th cells with regulatory function is identified Tregs. Treg subsets include thymically derived or natural Tregs and Tregs that are induced via post-thymic maturation. The two cytokines mostly associated with Tregs are interleukin-10 and transforming growth factor beta. Importantly, Tregs can themselves secrete these cytokines and use them to carry out their suppressive function. Tregs are immunosuppressive and generally suppress or downregulate induction and proliferation of effector T cells. For more information, please visit creativediagnostics.com.